In late 2019, a hitherto unknown coronavirus strain appeared in the city of Wuhan in China. It soon spread to other countries. Again and again, virus outbreaks threaten the health of communities and pressure researchers to develop vaccines. That's the case with many pathogens. Here's a little ABC about viruses. Some 3,000 to 5,000 species of viruses have been classified worldwide. Scientists believe that millions more have yet to be identified. Viruses are often transmitted by droplet infection. They're microscopic parasites that are neither really dead nor alive. They're not made of cells, don't carry out metabolic processes, and can't replicate outside of a host. When viruses enter the body, meaning when we become infected, they insert their genetic material into the host cells. The virus hijacks the cells in order to reproduce. When the host cell dies, thousands of new viruses are released, and the process starts all over again. In contrast to freeloading viruses, bacteria don't need a host to reproduce, and they perform generally beneficial functions in our bodies. When we are infected with pathogenic bacteria, we can beat them with antibiotics, right? Well, not necessarily. The problem is that bacteria are increasingly developing resistance to antibiotics.
Bacteria reproduce by cell division. Every so often, there's a replication error. Such random mutations can drive evolution via natural selection. Usually, they're of little consequence. The mutated bacterial form eventually dies out. In some cases, however, a mutation can result in resistance to antibiotics. And when antibiotics are used, the bacteria with the mutation proliferate. The antibiotic kills most of the bacteria, but the mutated bacteria are unaffected. The resistant bacteria survive and multiply. The use of antibiotics leads to a population of resistant bacteria. In hospitals, antibiotics are prescribed to many patients. That increases the chances that a resistant strain of bacteria will emerge. And patients sometimes bring antibiotic-resistant bacteria with them when they're hospitalized. Those superbugs are often washed into the wastewater system, and then into the environment. They end up on crops, in domesticated and wild animals, and eventually in us. It's a vicious cycle. Antibiotics are also used in large quantities in livestock farming. That also promotes the rise of resistant bacteria, which end up in the environment. New bacterial strains can spread around the world quickly, even within days. To combat the rise of superbugs, we need to understand how they spread. And we need to develop new drugs to combat them. Potent antimicrobial substances also exist in nature, wherever bacterial colonies are trying to survive and thrive, like in soil. An ordinary clump of soil is brimming with bacteria and fungi. They compete with their neighbors for resources and space. The microbes have a secret weapon in their arsenal. They produce chemical substances that can sweep away the competition. These antimicrobial compounds, or antibiotics, are of great interest to researchers. They isolate the microorganisms to grow them in the lab and extract the antibiotics they produce. In a screening process, disease-causing bacteria are applied to a plate. Then samples of the soil antimicrobials are added to the culture. The bacteria grow on the plate except where they encounter an antimicrobial agent that they're vulnerable to, known as a zone of inhibition. That's evidence that the substance is preventing bacterial growth in the zone. One day it could be the basis for a new drug.
And new effective drugs could come from a place like this, a mangrove swamp. Mangrove swamps are considered to be one of the most productive habitats on the planet, protecting coastal areas from erosion and providing homes to countless species, including a diversity of microorganisms. They're the object of studies by a young scientist from Indonesia. She's currently working at a research institute in Germany.
The development of new antibiotics is a race against time. Most antibiotics were derived from substances produced by soil-dwelling microorganisms. At the Helmholtz Center for Infection Research in Germany, Senlia Octaviana is studying sediment from mangrove swamps to isolate bacteria that produce antibiotic substances. The samples come from a nature conservation area in Indonesia. The microorganisms in this habitat have adapted to their tidal environment. To thrive in the sediment and brackish water, they've evolved to produce a variety of novel antimicrobial compounds. Could this be a new source for the drugs that are so urgently needed?
Sampai saat ini, seperti yang kita ketahui bersama,
We all know the world needs new antibiotics.
Membutuhkan antibiotik baru. Mengapa hal ini bisa?
A growing number of pathogens are developing resistance to the antibiotics used in medicine today.
Penyakit infeksi kebal terhadap
That's because so many antibiotics are being overused and misused.
Pemberian dosis antibiotik itu sendiri yang tidak tepat.
The biologist has been trying to find as many microorganisms as possible that produce antibiotic substances, and her work has paid off. She's already isolated 28 previously unknown species, mostly Myxobacteria.
Myxobacteria itu merupakan
Myxobacteria produce a large variety of antibiotic substances.
Tidak hanya antibiotik saja, kita bisa
Some of them can even produce compounds that are effective against malaria or viruses.
Myxobacteria itu sendiri, contohnya seperti anti-malaria, antivirus.
The Myxobacteria found in mangrove ecosystems have also attracted the attention of biologist Joachim Vink. He believes that Indonesia, which is home to about 1/5 of the world's mangrove ecosystems, could be a rich source of useful microbes. The research is still in its infancy, but Senlia Octaviana's project is a promising start.
Senli has done a very interesting work with this Myxobacteria. That is a special group of bacteria we are here working on. And she has isolated a number of novel species and characterized them, and is now on the stage that she can look for the bioactive compounds which are produced by this unique microorganisms. Indonesia is having a huge and wide biodiversity, and especially in the field of microorganisms, this biodiversity is not much investigated.
Senli Octaviana's initial findings showed that the Myxobacteria in mangrove swamps include many rare species. The bioactive compounds they produce will be screened to see if they have antimicrobial properties, which could help stem the rise of superbugs that have become a global health crisis.
Ya, surprise karena hampir sebagian
We were
besar-
very surprised to find that nearly every species we identified was a rare one.
Biasanya mereka hanya melihat di dalam kayak buku.
Usually,
atau
we only read about them in books or online.
suatu kebanggaan.
But here,
Jadi buat
we've been able to
saya,
work with them directly.
orang Indonesia, wah,
I'm
ternyata
very
kita
proud that Indonesia has so much potential.
luar biasa.
Senli Octaviana will soon complete her project at the Helmholtz Center. Then she plans to return home to Indonesia, where she'll continue her work on Myxobacteria and the mission to find new antibiotics.